What's up, everybody? Welcome to the Big Ideas Podcast, where we sit down with experts in science and discuss the big ideas behind their work. My name is Winter Beckles, and I'm very excited to speak with today's guest, Dr. William Browne, deep-sea diver, marine, motorcycle enthusiast, and developmental biologist. Dr. Browne is an associate professor of biology at the University of Miami, researching some of the questions surrounding how a single cell can eventually become an entire organism and the molecular and cellular mechanisms that control that process. Dr. Browne, welcome to Big Ideas.
Thank you.
How's it going?
Good.
I know you've been doing some training recently. Tell me about that.
So I for some reason, decided that I needed to do an Ironman triathlon before I turned 56, and this is the year.
Okay, so time's ticking.
Yes.
That's huge, though. That's awesome. When is the race?
It's November 2nd, so it's going to be the Florida Ironman triathlon.
Florida Ironman triathlon. And so there's a swimming portion, a biking portion, and a running portion?
Yes.
Tell me about those. What's the whole package?
Right, so the swim is, I believe it's 2.3 miles. The bike is 112 miles, and then it's a 26.2 mile run.
Biking 112 miles.
Yeah. So I just want to make it clear that my only objective is to finish.
Get past the finish line. Yeah, I have not yet put myself to that challenge, but maybe one day, yeah, I'll be inspired to do it. I'll watch your progress and maybe I'll catch some of that juju. But how does a scientist also have time to be Ironman? So tell me a little bit about how that. Yeah, what does your day tend to look like?
So it's not easy. I do my science, and I also have teaching commitments that are sort of fixed. So what I've ended up with is sort of a lopsided schedule where I'm in the lab until relatively late in the evening / early morning, and then start off my day with whatever the training plan is, and then teach, and then back in the lab.
So training in the morning, teaching midday, mad scientist at night kind of thing?
Yes.
Yeah, very cool. And then I think when we talked about this before, but you say sometimes you're here till 1, 2 a.m.
But it's fine, right? Because it's not really work for me. I love it. So I don't have a problem staying extremely late.
Nice. I mean, the building's dark, building's quiet. Do you pump up some music? Do you turn it into a Dr. Browne party, or is it more tame than that?
On occasion.
Yeah?
Yes.
I like that. Good. Now, I would love to get into the big idea driving your work. How would you explain that for us?
For me, the big idea, the big driver is what are the fundamental characteristics of a metazoan or an animal? Right? And when did those characteristics arise during the diversification of animals?
Okay. And you have a really interesting system. I want to give you the chance to explain how you came to work with this system and what are the questions you're asking. And I've got that video, so whenever if you'd like, we can pull that video of your system up to reference it.
Yeah, you can pull it up whenever. I guess I can start sort of at the beginning. So it's nice to mix a little bit of adventure in with the intellectual questions. So the model system that I have in the lab, it's an organism called ctenophore, and they're these gelatinous animals that for most of their life are living in a pelagic environment, so in the open ocean midwaters. So how did I stumble upon ctenophores? My PhD was based on developing a crustacean as a model system for thinking about development in the context of an amphipod called Parhalate hawaiiensis. And there's a whole group of these amphipods that they're called Hyperion amphipods. They also make a living in oceanic midwaters. So after I got my PhD, I moved to Hawaii with the objective of sort of figuring out the evolutionary history of these midwater amphipods. So I was doing a huge amount of diving and oceanographic expeditions and working with ROVs to to sort of understand the context of oceanic midwaters and also collect the animals that I was interested in. And it turns out that a significant number of these high-period amphipods associate very closely with ctenophores. And ctenophores are just, they're stunningly beautiful, right? So very quickly, I decided that sort of the next phase of my research career was going to focus on ctenophores. So just beyond their sort of aesthetic attributes, one of the important things about the group for me as an evolutionary biologist is that they diverged very early from other animals. So they present themselves as an interesting group for thinking about both characters that are fundamentally associated with what we consider to be an animal, and then as an evolutionary biologist, the attributes that are explicitly associated with the trajectory of that lineage through time. So ctenophores are likely a lineage that's been separated from other animals for 700 million plus years. So very, very ancient lineage. And because they're living in this unique environment, they have a lot of adaptations and character traits that we're not really used to thinking about, right? So again, a useful group for thinking about what makes an animal, and then what makes ctenophores unique from other animals.
And not too many model systems can be used in that way. Is that true? Or?
For thinking about sort of the context of universal traits associated with all animals, you really need to be looking at least one very, very, very old lineage.
Okay.
Right? And so there's been this sort of contentious debate the last couple of decades about whether the ctenophore lineage represents the oldest extant group of animals versus sponges.
Okay.
A lot of back and forth, right? And, for most folks, for the questions that they're interested in, it honestly doesn't matter. They just need information from one or the other lineage in order to polarize their observations.
Okay.
But for this other smaller group of scientists that are interested in early evolution amongst metazoans or animals, it's a more important question to consider.
And how do you and your team go about investigating these things? What are you guys doing in the lab?
Yeah, so we're trying to apply as many useful tools as possible for thinking across many scales. Right? So there's the organism itself, which is a collection of differentiated cell types that are all working together to allow this animal to develop over time and then contribute to the next generation. We're also very interested in better understanding of those individual cell types. So most of the effort in the lab right now is directed at developing a better understanding of context for the innate immune system for these animals by generating cell cultures and identifying and isolating putative immune cells. Right? And then to consider their genetics. So we've got whole genome from these organisms, and we clone genes, and we use a variety of functional genetics approaches to query gene function.
Very cool. And again, this goes, I think you mentioned, back to examining properties of their immune systems. Specifically?
Right now, yeah. The big driving questions in the lab are, now that we've established that they do have immune cells, it's a consideration of the range of immune cell types.
Okay.
Right? So do we have immune cell types that phagocytose pathogens? Yes, we do. Do we have immune cell types that engage in other behaviors associated with immune cells in other model systems? So, for example, contrasting against mammalian model systems where, pretty much a wealth of our knowledge as biologists comes from. And the really sort of interesting and I think eye-opening thing is that you can't get further away from what a human is.
Yeah, so let's talk about this a little bit, because this thing is really beautiful and bizarre, and as you're saying, completely other end of the spectrum of what I think of in terms of an animal. So humans over here, these things are over here.
Yeah.
How does
Yeah, but the cell types are there. The cell behaviors that we know exist in humans or other model systems, mice, rats, those cell types are present in this animal, which suggests that, there's some very, very fundamental set of cell types that were present extremely early in animal evolution.
You were describing last time how some of these cells are moving in these petri dishes and how you can kind of identify when you have a culture of cells, which ones are worth further study versus which ones are not. Can you break that process down for me? It was really cool.
Yeah. So it's guided by what we know, right? For example, in the context of a search for immune cells, classic descriptions of immune cells are cells that move around within the organism, and if they recognize pathogens, the typical behavior is that they will swallow the pathogen and then break it down. So what we were looking for were, number one, cells that were competent to move around, so motile cells. Then number two, whether or not those motile cells would recognize potential microbial pathogens and respond by phagocytosing them or eating them, and then sending those to a lysosome to be broken down. So again, this involved developing cell culture approaches and then phagocytosis assays, which sort of leverage our ability to fluorescently tag microbes with a pH-sensitive tag. So the tag changes color based on local pH. So if this microbe is phagocytosed by a potential immune cell and sent to a lysosome, the lysosome is acidic, and then all of a sudden you get this production of, and depending on the tag, either green light or red light, and that identifies that cell as a potential immune cell, which for us means we need to get more of these because these are the cells that we're potentially interested in studying further.
That's really cool. So essentially you have a petri dish with cells from the ctenophore, and you're introducing pathogens that will let off a little signal, more or less.
Yeah, they'll glow if they've been eaten.
That's insane.
Right, and then they go from there, they go through a machine that's an extremely useful tool called FACS. This is fluorescence automated cell sorter. So we've got our cells of interest that are now glowing, right? So we can use this microfluidics approach to collect just the glowing cells, right? And now that we've got a whole bunch of these glowing cells, we can do additional assays, ask further questions about behavior, or take those enriched cells of interest and do some of the genetics experiments that we're interested in. So sequencing subpopulations or sequencing at the single cell level.
Because every cell, although it has the same genome,
it expresses different genes, right?
And so an immune cell or any type of cell is going to have the expression profile specific to that type of cell.
Yes, so they'll have, an immune cell is going to have an immune cell type expression program. There are many, many, many different immune cell types, and they all have discrete gene expression programs associated with them. So we can take our collections of cells and ask, do we have just one general program associated with immunity? So effectively one immune cell type as far as we can tell from gene expression? Or do we have discrete gene expression programs that are either unique to ctenophores, which might represent a unique cell type associated with ctenophore evolution, or are these different programs associated with immune cells, are they similar to gene expression programs that we see, for example, in our immune cells.
Gotcha.
Which would, of course, indicate, a very, very old,
Highly conserved
immune cell type.
That's incredible. So you're looking essentially at gene expression of individual genes in an individual cell or a group of cells within an individual organism, and from that asking questions about processes that did take place millions and millions and millions
Hundreds
of millions of years ago.
And there are inferences, right? Because we can't be there, but you can have weak inferences and you can have strong inferences, right? And so the effort is to confidently identify patterns that strongly infer one hypothesis over another.
Very good. How did you end up doing this? I mean, growing up in late 70s, early 80s, Southside Chicago, naturalist almost at heart from what you've said.
I was a kid, and we would go on these trips over summer vacation, and my mother got me a guide to insects for wherever it was that we were going each year. And so what I would do is, to her chagrin, once she realized what she had done, I would collect insects on our trips and then identify them and put them in cases and run around the neighborhood and collect.
Look at my dead bugs.
Yeah, look at my dead bugs. Wow, great. Right. So that's where, I started, very young and innocent. And then, it sort of faded away for a while. But it was always there. So I ended up in the Marine Corps after high school. And that's where I started scuba diving. And one of the things that, and this was in Okinawa, which has just really beautiful coral reefs. So I got really, really into diving on these coral reefs, which sort of often didn't fit with exactly the job profile at the time.
So you're out there training for the Marines, and you're diving for work, but you're loving the coral reefs there. You're seeing the biodiversity. I mean, I've seen coral reefs, but could you describe what you were seeing to us?
So interestingly, the bombing campaigns from World War II generated lots of sort of pockets of what I can best call coral amphitheaters. So it's just big exposed stages with these walls of coral all around you. So it's just teeming with life and lots of things that, of course, I hadn't run into on the south side of Chicago. Like sea snakes were really abundant. Yeah, so it was very eye-opening to be have the opportunity to be on coral reefs on, at times almost a daily basis.
That's beautiful. And you still dive?
Up until about 4 or 5 years ago.
Okay.
Yeah, so once I got Nemiopsis, our model ctenophore, into the lab, dive-based field work became less of a priority than the development of the tools to explore the biology that you were interested in.
Gotcha. Wait, so you were trained to dive in the Marines as a Marine, and then carried that forward and used diving to collect your samples?
Yeah, so I used the diving during my PhD. I would go to Smithsonian field stations, both in Panama and Belize, to do collections of the crustaceans that I was working on at the time. And then once I got to Hawaii, just because of the profile of the hydrology there, you can get into very deep water very quickly. So I started doing a lot of very deep diving, using rebreathers for a few years during my postdoc. That's where I really got the bug for ctenophores.
Yeah. I mean, that's not hard to believe. I mean, I have this video here. Can you walk me through? I won't even try and describe it. I'll put the video up on the screen, but what's going on here? Yeah,
so ctenophores, they're named for these teen rows. So ctenophores have 18 rows around their biradial body plan. Those teen rows are composed of modified giant cilia arranged in paddles, and these paddles beat metachronically to sort of propel the animal through the water. So they're the largest animals that locomote purely via ciliary action. And those paddles function much like a diffraction grating. So if you shine white light on them, that white light will be broken up into its component wavelengths. So it generates this really attractive rainbow pattern.
Yeah, you get sucked in. You watch it and you just want to keep staring.
Yeah, it's rhythmic.
It's just very rhythmic, meditative almost. And so you're down there to study crustaceans, but you keep getting distracted by these comb jellies. And now you study them. How do we make that jump, though, from military to studying science?
So I had a training accident in the military, which cut short that career path. So I came back to Chicago and enrolled at community college. And within a few months was introduced to the curator of fishes at the Field Museum of Natural History in Chicago. And as a kid, one of the things I did, other than collecting insects, was to have fish tanks. So that was one sort of thing that my mother was sort of happy to let me.
A fish tank is an easier sell than dead bugs.
So fish tank, they're contained, right? So they're not going to go anywhere. So she let me set up a few fish tanks around the house. What I ended up doing, since I had more than one, was to set up sort of these different sort of eco-type themed aquariums. And the one that I was most proud of was the blackwater ecotype from South America. So this sort of Amazonian ecotype with sort of an acidic water profile with a lot of tannins in the water and dominated by carassiform fishes, so tetras, and a few other interesting fish species, but mostly tetras. And the curator of fishes, when I initially met him, one of the things that museum fish collections have, in addition to lots of jars of preserved fish, is that they've got lots of fish skeletons. And so he opened a cabinet that was full of cleaned fish skeletons, pulled out a drawer with this massive skull from what's called a pacu, which is a South American carassiform fish. It's in the same family as piranhas.
Okay.
And he asked me what it was. And I knew what it was, but that was the first time I had seen one that large. So I guess my eyes got really big and I must have inhaled sharply. Exactly. Oh, that's a pacu. Right. And so he hired me on the spot because I knew what the fish skull was.
He saw it in your eyes, like this kid's got it.
Yeah. So he let
How
me volunteer.
Did you know?
So skulls of vertebrates are packed with unique character traits. If you're going to look for synapomorphies that are going to be associated with any particular species, often some attribute of the skull is going to be a giveaway. So there were sort of general patterns associated with the skull that placed it in the same group as the piranhas, but these pacu don't have sharp teeth, so they don't eat flesh. They crush seeds. So their dentition is highly specific. They've got these massive molars that are designed to break open seeds during the wet season. And in the wet season, they're actually swimming through the forest.
So this is very specific.
Yeah.
Skull he shows you, and you immediately had it.
Yeah.
That's neat. I mean, again, so maybe not from birth, but you kind of had this in you, and it followed.
Yeah. It was always there. I just didn't know as a teenager, I didn't know that was a career path that was open to me.
So he hires you on the spot as a volunteer?
Yeah.
And what happens there?
So he gave me lots of interesting tasks with regard to sort of being able to identify different species of fish, again, based on their skeletal features, general morphology, and I very quickly developed a sort of a nice set of skills revolving around identifying crassiform fishes. And he, at the time, was going on several expeditions every year to Central and South America and literally bringing back 55-gallon drums of fish.
No way.
And he can't identify them all, right? Especially with duties associated with being a curator. So that once he was comfortable with my ability to sort of objectively identify things, then he hired me. It became a paid position where I was identifying all of these fish that he was bringing back from his expedition. So probably my longest term legacy are going to be, these jars of fish with little labels that say identified by William Browne.
I love that. Yeah, you're leaving a little signature. How long was that stint then?
That lasted for several years. So that lasted through, I transitioned from community college to University of Chicago, which is where I got my bachelor's degree. And that position lasted all the way through. Now, I have to admit that at some point molecules became really the driver versus very focused systematic or cladistic approach based on looking at a whole organism and looking at morphological traits. And honestly, that in part was Barry's doing, because he was assigning me groups of fish to work with that were extremely difficult to sort of bin into one species or another species because they had a lot of very labile character traits. So at some point, it became clear to me that the only way to solve some of the phylogenetic questions or systematic biology questions that I was being presented with was to look at the molecules. Right, and maybe there's something with regard to sequence variation that would allow one to, with confidence, say this group of things are distinct from this other group of things. So my last year and a half of my bachelor's, my undergraduate tenure, I was working full-time in the fish department, but also full-time in a bacteriophage lab.
Nice.
Gaining some skills working with sequences. So that's where I learned how to clone genes and do sequencing reactions.
So you're starting with organization at the large scale, the species scale, the individual scale, but the organization's happening at a much finer level now. And so you had to get a whole new skill set to tap into that level of analysis.
Yeah. And the bacteriophage projects were focused on identifying promoter structures and thinking about polymerases that bacteriophage was using to transcribe its genes. So very, very disparate, but I brought them together in my PhD in the molecular genetics and cell biology program at the University of Chicago by, again, trying to identify a crustacean that we could use as an additional arthropod model system to validate what we thought or presumed were general rules with regard to arthropod development that were almost entirely based on experiments coming from Drosophila.
Wow. And this is, if my timing's right, circa 2000?
The mid to late 90s.
So this is, you're really at kind of the leading edge of some of these techniques as far as being widely used across research institutions. Like sequencing back then wasn't what it is today.
Yeah. The sequencing reactions were not fluorescent dye based; they were radioactive based. So it was a lot, and those were the film days.
I've heard about.
So you had to develop. Number one, you were like making these huge polyacrylamide gels, right?
Fun.
And then running many, lanes of these sequencing reactions where you're introducing nucleotides that had a radio label associated with them. And we develop them in the dark, so as your radioactive nucleotide is decaying, it's like producing a little bit of light, which is going to develop that band on the gel.
Old school.
Yeah, and then in order to read the gels, you would have to, like, the best practice was to have like two folks read the gel at the same time so that you could catch
Each other
making mistakes as you actually read the gel.
So not nearly as foolproof. Well, today's methods aren't foolproof either, but it was a lot more of that sort of manual checking, human error sort of.
Yeah.
Before you got handcuffed to the bench, did you have a chance to go on any of those expeditions with the PI at the Field Museum?
I didn't go with the PI at the Field Museum, but he recommended me strongly as someone that needed to be a part of an expedition that was being led by a couple of postdocs and some graduate students in the Amazonas province along the border of Venezuela and Colombia, right? And so it's this really interesting expedition where the focus was on a group of fish called gymnotiforms, which sort of interpret their environment by reading magnetic fields. And we spent a lot of time along a Blackwater River called the Atabapo River, which runs south and ultimately ends up feeding into the Amazon. And at its sort of north end, it is joined by a river coming out of the high Andes in Colombia called the Guaviare. Okay? And so where this whitewater from the Guaviare that's loaded with its high pH, high hardness, a lot of minerals and signature coming out of headwaters from the Andes, meets this black water. There's basically a shower of particles coming out where these two rivers sort of join and intermingle. And these gymnotiforms make use of that environment, which is extremely problematic for any organism that's orienting based on vision.
You can't see through.
You can't see it.
Gotcha.
Right? But if you can read magnetic fields, then it's okay.
Okay.
Right? So they would literally hang out. There were a number of species that we were interested in collecting that would hang out in those outfalls, right where the two rivers met, right? And then there were a number of species much further south that were restricted to just the black water or high acidity water associated with the Atabapo that we were interested in. Most of the folks on that expedition were not familiar with carassiform fishes, but carassiforms dominate the sort of the fish population.
And those are the fish you were drawing, right, at the Field Museum. So you're the guy.
So I was the guy who was identifying all of the fish that they weren't actually focused on.
Okay. Hey, I mean, it got you a ticket to Amazon.
Yeah. So with those kinds of expeditions, it's even the way that things work now is that if you have an opportunity to go to an understudied area, even if what you're focused on is just a handful of species, you devote some time to sort of a broader collection so you can have a broader context for the general environment. So yeah, we brought back several 55-gallon drums of fish from that.
How are you getting around with?
So we were using gill nets where we could.
I'm not familiar with that.
So these are nets that you can just place in a current and catch.
Functions like a gill, like a cotton. Okay.
Yeah. The other thing that we were doing for, particularly where the Guaviare and the Atabapo met, the water was actually quite deep. So we were using a trawl net, which requires using a fairly heavy boat. Right, so that you're dragging the net and the net's not dragging
Catching you and pulling. Oh man.
Right. So, but this is also, very remote region of Venezuela. So what we ended up with was a dugout. I wouldn't really call it a canoe, right? So this dugout boat, it was from a single log. It was probably about 70 feet long, very long. The widest point on the boat was maybe 3.5 feet.
Oh, yikes.
The rear third had sort of this thatched canopy on it. And we lived in that boat for 3+ weeks. And there were 7 of us total.
How many
and
people
there was
fell
plenty
in?
of room.
Really?
Yeah,
It's
there's
spacious, and it's how does that?
Well, it was spacious in one dimension
Okay.
and not so much in the other dimension. Right. So there's just enough room where 2 people can sit across from one another and have a little bit of workspace. There was plenty of places for example, for our 55-gallon drums. So we had a couple of 55-gallon drums. And it did have a motor. Right.
My goodness.
Yes. Which is this whole other logistical issue.
But when things are going well, you're just cruising down and just having this amazing view around you, right? Like,
Oh yeah, yeah.
it's just-
So
what did you see while you're out there?
So there were literally pink dolphins. We were eating piranhas almost daily from one of our meals.
How is that? How is that? How is
tastes
piranha?
awesome.
Yeah.
Yes.
I one day. Like
And swimming with them, right? So piranhas are not, despite the horror movies, unless you're bleeding, piranhas aren't an issue.
That's cool. Yeah.
But one of the graduate students, he was an ornithologist. And so the thing that I didn't appreciate prior to this trip was that when you're in a multi-story canopy and you're following birds, you're not necessarily seeing them. You're just listening to bird calls and following bird calls. So when we would finish whatever we were doing on the river, that's when what he had been looking forward to doing, he could then go do, which was to go off and follow birds and see what was out there. And so one of the things that ended up falling on my shoulders that trip was to make sure that I got the ornithologist back to the camp. And so he would be following bird calls, and I would have to keep track of where we were and how we were going to get back to the camp. And these were not sort of directed walks. They were walks following wherever the birds that he was interested in were going.
Just stochastically through.
Yeah. So
brush.
they were all, I wouldn't say that they were random walks, but they were pretty close to random walks. So
How do you not get lost in that situation?
right. So particularly in a heavy forested area, you can take 5 paces and then turn around and look behind you and not recognize the space that you just walked through. So there was a lot of referencing what the terrain looked like and trying to identify features, and then leaving marks on readily identifiable objects that an animal wouldn't make. So making cuts on trees or breaking branches and laying them in ways that, even if something large came through, it wouldn't break it and lay a branch in a particular way, just so that I would have landmarks to follow back, because the way back was often, literally a recapitulation of this very nearly random walk to wherever we had gotten to. But it it was interesting. But the region is also known for Permian granite pushing through, so uplifting of granite. So the terrain was a lot more variable than you would assume just based on the vegetation. You could hit like very steep stone outcroppings and things that were below the canopies, right?
That's bizarre.
So yeah, it was really interesting.
It feels like a cliff, but it's almost in
It's a
miniature.
cliff, but it's surrounded by like green.
Yeah.
Right? So
You're walking into walls literally while you're going through this.
exactly. Exactly.
That's nuts.
It was my sort of first scientific expedition, so it was all new for me.
Okay.
So I was
like, hey, this is
fine.
it.
Yeah, I was fine with it. And I just, tried to contribute, 100%, 110% of what I thought I could do to make things successful. And, there were points in time where, I was a hindrance for reasons that no one would have predicted beforehand. Because, it looks like I could have been from Venezuela or Colombia, which ended up being a problem going through checkpoints. In these rivers, literally, you'd have checkpoints, Venezuelan checkpoints on one side of the river, and then you would occasionally run across Colombian outposts on the other. We had papers for this multinational approved expedition that we were on, but, I didn't know it would have been better if I hadn't been wearing my old military fatigues on this trip, which is what I packed to bring with me, right? I couldn't speak Spanish, right? So who's this oddball guy who looks like he could be Venezuelan, but really not, and can't speak the language, and
Spy vibes.
you guys got to hold on here at this checkpoint while we check and make sure that things are exactly what we think they are.
Okay.
So
Overall, just suspicion.
yeah,
And no one else on the team.
would have known that ahead of time, because most of them were sort of neophytes as well. We were all young,
But
yeah. Our liaison knew. He was like, they're following up on you, Bill.
it's not so random.
It's your fault.
So couldn't you learn some Spanish for us, Bill? Just for this trip.
Why did you bring all?
I guess it's the military gear. Yeah, no, but you had, I guess, multiple outfits of that. You couldn't just not wear that on those days. You're like, this is all I brought. This is my gear, guys.
Yeah, it's like, because I did most of my service when I was in the Marines in Okinawa, which is subtropical, and I also did a couple of exercises in the Philippines. So it's like I was wearing what I thought was appropriate for the climate where I was going to be.
Absolutely. No, I don't know about you, but I think sometimes the gear is one of my favorite parts of biology. Like sometimes you get the cool equipment, or if you're in these extreme environments, you need special boots or special. So I always feel when we're packing up, all right, I'm getting my stuff together.
Yeah,
but
it's
then
fun. It's the fun part.
it's super fun, right? And then I have do you have favorite things that you kind of try and find use for in your civilian life that you can kind of still use? Or
Yeah.
there you go. What do you have? Let me hear it.
So for following the local coyotes, I set up lots of camera traps.
Down here in Miami?
Down here in Miami, because initially no one believed me, because I was running through a patch of pine rocklands. I don't know if I ever told you this. No, it's running through a patch of pine rocklands, and it was raining, so there was a fair amount of sort of environmental noise. And I came around a corner where I couldn't really see well because there were a bunch of palmettos and slash pine. And I crossed the path with like the what I would figure out later was the alpha male of the coyote pack. Right? And he was pretty big. Initially, I thought it was maybe like a red wolf. And then I did some research and was like, it's probably not a red wolf. It's probably a coyote.
It's like Labrador sized.
Yeah. Yeah. Yeah, with yellow eyes.
Terrifying.
Yes.
That's
yeah,
scary.
I think we've got sort of this innate response to mammals with binocular vision that have yellow eyes.
It means run.
Who has yellow? When you have binocular vision as a mammal, who has yellow eyes?
Predators.
The predators.
Run. Or don't run, right? Like, what? Yeah, I guess. Teach us, Bill. What?
Well, I yelled, and
Yeah.
he froze as well.
Okay.
And he didn't want to give up space to me initially, so we just stared at each other for, it was probably maybe 3 or 4 seconds, but it was long enough.
Yeah.
Right? Then he sort of like trotted off, and Michelle was actually with me. She came around afterwards, and I was like, let's backtrack and get out of here right now, because there's a wolf or coyote. So I was flying drones around on, like, I would program a pathway and just like launch them, take off and fly over. I was trying to catch one from above. And then I set up a whole bunch of camera traps. I should send you a couple of pictures.
You absolutely should. It sounds sweet.
Yeah, so the pack is, there's more than one pack now, and they're actually pretty large. So the main pack is in the USDA land.
Okay. And they didn't know about it? They were unaware? Or it just wasn't really public knowledge?
So they weren't aware. So I think the USDA, there's no way that the folks running the USDA facility could not have known.
Okay.
But Florida Fish and Game did not know until I showed them pictures. They were like, "Oh, it's probably like a stray dog." And I was like, look at these pictures. This is
This coyote-looking thing? So you see this and you're switched on. You're like, all right, bring out the gear. Let's figure out what this is.
yeah,
I like that. Pull the toys out.
yeah. So I've got now years of like footage. I know their patterns because I know it was a little bit uncomfortable running across them. But now I see them in the neighborhood. I'm out walking the dog in the middle of the night.
And over here, because you live 10 minutes from here?
7.5 miles, not that far away.
Okay.
They're definitely in Pinecrest at this point.
I feel like, is that...
So I occasionally see them on my motorcycle, right? Because my motorcycle's quiet, so...
Oh, because it's electric?
Yeah.
Oh, that's sweet.
Yeah, so they don't run until, I'm close enough that I can actually see them. And I go, what's that weird buzzing sound?
That's bizarre. This is your first electric bike, right? But how many bikes have you had? And what were their names? 3 bikes.
I had a Honda CB750. That was the first time I broke my leg. And then for graduate school, I wanted something that reminded me of the Marines, so I had a KLR 650, which is like
I'm gonna look it up.
the old sort of military bike. I had many adventures on that.
Were you riding bikes in the military also? Is that...
No,
It just
but I always wanted to. I felt like that was something that I missed out on. I wouldn't be surprised if they're switching over to e-bikes and electric motorcycles now.
I mean,
They're quiet and they don't have a
that's
heat
stealth.
signature. Combustion vehicles have a gigantic heat signature. And even when I was in the Marines, what I was most often doing was being assigned to set up an observation post and take the optic system from a tow missile system, a vehicle-mounted tow missile system, get dropped off where we could sort of get some high elevation vantage point. There was either a road below us or but there was going to be some activity that we were going to be observing and then reporting back to the battalion commander. So we'd find a site and set up the thermal sight system and just sit on it for like 3 or 4 days and just watch. Any vehicle that comes through just shows up like it's on fire.
you're up there on the cliffs looking for heat signatures of anything.
Yeah. During the day, you use normal optics because the light's fine, but that really is the most useful at night, because if you're producing any heat above ambient background, it stands out like a sore thumb.
That's cool.
So these days, the way to get around it is with electric motors, because There's no heat signature at all. I ride my bike on a daily basis over 100 miles per hour. And the motor gets up to like 150 degrees, maybe.
At 100 miles per hour?
At 100 miles per hour. For like,
sustain.
I can go sustain for long stretches, and the heat signature is tiny.
Does it read that to you, the output of the engine temperature, just as a, so you're like...
Yeah, toys, man. It's like communicates to my phone. I've got my phone mounted on the handlebars.
Is it in your ear? Bill, your current engine temperature is 150 degrees Fahrenheit.
Gotta be the next toy.
Oh, I love that. I'm going to come see your bike pretty soon. I want to see this. Where are your favorite spots to ride in Miami?
Oh, I just commute. I don't.
Oh, that's your...
Yeah, I don't ride for fun because I know my limitations.
Gotcha. Okay.
Yes.
I can respect that.
Yes.
Just to and from work. Is there anywhere you would love to ride if you could ride? Because down here,
well, my ride's pretty good to and from work.
I was going to say that. So is that where that 100 miles per hour?
Yeah. So it's right next to the USDA with the coyotes. Right? It's a good
just stretch.
Yeah.
Nice.
Yeah, and so that's where lots of kids with overpowered cars take their cars at night. Stretch. So
it's the place to go. Test out my electric motorcycle.
There are more than a few Corvettes and Porsches in the neighborhood that make use of that stretch. There's some kids with some way overpowered Mustangs that I'm sure they're holding on to those things with just dear life, man. Please don't crash.
No. Yeah, you're like, it's cool up to everyone watching, but inside you're just like praying that you keep it straight.
Yeah, it's a perfect like Tesla stretch.
And I've never seen one of those let loose, but every now and then I see one like zigzag through traffic, and you can tell they're quick. I can just imagine an open stretch.
It's like a motorcycle. It's like instant power.
That's kind of scary, though. I guess that's the point. Well, that's not the point, but if you're getting a car like that,
it's super helpful. If you understand how to use it, it's super helpful for keeping you out of trouble.
You can stop immediately, or be around something almost immediately.
Yeah, I mean, if something is happening around you, you don't want to slow it down, because the probability is that you're going to drive into whatever happens.
Gotcha.
You want to get ahead of it, so that if something happens, it falls even further behind you.
Smart.
So counterintuitive. Speed up to stay out of trouble.
Yeah, I can see it. My older sister, she I think has even taken the test to get her motorcycle driver's license. I don't believe she passed. We'll edit that part out. But she's always wanted to, and I've always been worried. I'm like, oh yeah, it's super fun, but there's that danger. But I think that's part of the appeal. So
yeah, but like I very rarely ride on US1 because it's just too dangerous. But I don't ride on the 826 or the 836 because there's always someone blowing by you at over 100 miles an hour.
Yeah.
Fully protected, right?
In a steel box.
Yeah, if you wipe out on a motorcycle at, anything over 40 or 50 miles an hour. Both times I broke my legs, it was sub 30 miles per hour. The first time it was like 10 miles per hour, if that.
Have you had other injuries? I know you had the accident, but aside from that, do you have other hardware in you? Are you a true Iron Man, or are you...
No, I just have pins and plates from the motorcycle. So I've got plenty of scars. It's all soft tissue stuff.
That's good, though. I don't know. I feel like sometimes sports, motorcycle, these hobbies, they are a little dangerous, and then you live through any accidents you have. But at least for me, when I've been hurt, I tore my ACL a couple of times, and it makes you pay attention a little more. It's like your mortality. All of a sudden, it's like, ooh, okay, yeah, that hurt, and I'm okay, and I'll get over it. But I'm gonna slightly adjust these rules I'm living by just a little bit for that, scenario or whatever.
Yeah, I was in a helicopter once and had one of those moments. So the helicopter was in an old UH-1 Huey, and I was cargo. So it was a training run for the student who was becoming a pilot. So it's part of his helicopter pilot's license, and they were doing, it's like a half-day thing. So refueling, landing at a couple of airports, and then doing some NOE maneuvers, or nape of the earth maneuvers, and that's like you're right above the tree line.
Okay. Navigating super.
So
is that super cool? Yeah. Terrifying, but like.
But in a good, it's not,
your adrenaline.
So one of the other folks I was with was clearly terrified. I thought it was great. It's like, a real life roller coaster.
How fast are you going in this thing?
It was pretty fast.
Yeah.
It's pretty fast. But when you have the side doors open on a Huey, you've got these out-facing seats. So there's like 2 or 3 seats facing the door on one side, 2 or 3 seats facing the door on the other side. Doors are open, tons of noise, tons of air movement. We had on these headphones so that we could hear the pilot discussion with the instructor, so we would know sort of a little bit of what was going on. And then there's this harness system that you lock yourself into. It's a pretty straightforward harness system, and clips and things. And as long as you're putting pressure on the system, it definitely locks in place. And so he's doing these maneuvers, and I'm loving it. So I'm leaning against the harness so that when the rotation is on my end, I can look down into the trees.
Yeah.
Yay! And when we got back, I sort of relaxed and leaned back, and the whole harness just fell off. So I had not clipped it, and no one checked to make sure it was clipped. So if at any point during those maneuvers, if I had relieved pressure from the harness system, it would have opened and I would have fallen.
Jeez.
So that was a mortality check. It was like,
that's that moment. Holy crap, I almost died.
yeah, that was great. That was a lot of fun.
You're like, woo! And then 3 seconds later, it clicks. That's crazy.
Well, it clicked after we got back.
Oh, so it's not until your
Flight was over, and I
that's when you finally relax.
I relaxed, I leaned back, and the whole thing it's like, oh my God.
Did you lock eyes with anybody?
No.
This is a private thing. It's happening
It was a
inside
private
of you.
thing. It was a private, because it was a training flight for this. I've got in trouble more than once. It was the cargo was unsanctioned. The flight training was sanctioned.
Okay. And you were the cargo.
The flight training was going to happen. The additional cargo was not on the books.
Gotcha. So your participation.
Right.
So you couldn't then say.
No, it's the worst thing that could. Yeah, the worst thing I could have done was
hey guys.
Oh,
I didn't. By the way,
my harness was not secured.
No way. No, you got to keep that one to yourself. That's crazy. And then so I guess that's one of those moments where it's like, okay, that was fun, but next time.
Yeah.
It's intense. Sounds fun though.
Yes. The other good mortality check is skydiving. Not tandem. You have that check when you leave the plane, right? Because under your own power, you're leaving the plane. If you're into that kind of thing, that's one of the ultimate mortality checks. All right.
That second before you jump, is it in there already, or is it still fun and adrenaline?
No, that's the hardest part. The hardest part is to exit the door. right? Because you're good. There's some degree of safety associated with being on the plane,
Right.
no matter how rickety the plane is, right? Once you leave the plane, you're completely dependent upon this nylon parachute deploying like it's supposed to. Yeah, but ironically, it's like way more peaceful than being on the plane. Once the chute opens, it's the most peaceful thing.
Yeah.
And that I've like ever experienced.
Interesting.
Ever. Right? Because the sensory input is completely different. So once the chute opens, there's not a lot of wind noise. There's just a little hint of noise coming from the ruffling of the chute above you. There's no surfaces to reflect noise back. So you can shout as loud as you want, and it sounds like you're whispering because it just goes away from you. It never comes back. So it's pretty amazing.
That's awesome.
And you're sitting in like a cradle. So you're traveling with the wind, right? Wherever the wind is going, you can steer against the wind and pick up a little bit of wind noise, but mostly you're a passive carrier on the wind.
That sounds so serene.
You're a seat on the wind.
I love that.
But until you open the chute, it's violent as hell.
Are you? That's the part that's the part I think of. I never thought of the after the chute part. I just think of the, holy cow. So you mentioned the feeling of peace once you pull the chute. And it makes me think of what I've heard divers describe when they're underwater. Now, is it similar to that experience?
Yeah, except the feeling of weightlessness is greater when you're underwater.
Okay.
Especially if you're using a rebreather. Yeah, so a rebreather isn't responding to changes in tidal volume of your lungs because you're not exhaling the gas. You're recirculating the gas. You're not going to go up and down in the water column based on breathing characteristics. So it's very much like being in a weightless environment. I've also been a part of oceanographic cruises that were focused on using remotely operated vehicles. And for science, remotely operated vehicles are definitely the way to go. Not a human-occupied vehicle, because the primary mission for a human-occupied sub is to bring you back alive. So the science is already secondary. So you don't get nearly as much done. Whereas for a remotely operated vehicle, if it were to fail at depth, it would cost a lot of money, but it wouldn't cost any human lives. And you can also dedicate all of the available payload to executing whatever the science mission is.
What's the coolest thing you saw while you were down there?
Dinosaurs.
Yeah. That's what the trips were for?
So overnight we would do trawl casts. So put a depth meter on the trawl, run the trawl to depth, open it up, collect bait for a few hours, bring the trawl back up. But that's only going to really be efficient for bringing back things that have hard parts, because it's a net and it's moving through the water at whatever speed the vessel is moving. And then if the conditions are right, then midday we would do a blue water dive. So a team of us would get scuba gear, get a rib, deploy the rib from the larger mothership or oceanographic vehicle, and then take the rib about a mile away. So just to be able to see the vehicle on the horizon, and then do a blue water dive for about an hour or so. And that allows you to collect things from the surface to about 80 feet.
Okay.
So that sort of tops the ice. Right? But, if you're in a water column that's 6,000 feet deep, right, that top 80 feet, it's interesting, but it's not telling you anything about the other 5,950 meters.
Yeah.
Well, that was one of the benefits of being in Hawaii. You could do a blue water dive over, 4,000 meters, 6,000 meters, but still see Oahu on the horizon.
That's cool.
So you could take a 30, 40-minute boat ride out and be over significant column of water.
Deep water.
Yeah. And those are cool. Blue water dives are cool. You're like on an upside down trapeze, so there's a float at the top, right? And then you drop a line with usually a weighted section of chain link at the bottom, so it sort of drops straight line, and that's going to be the thing that determines their loops every 10 feet or so. So that's how you can sort of set a particular depth and stay there. And then each diver is on a 30-foot leash, and you attach that leash to a little ring that's hooked onto the loop. And you've got this centroid where all the divers are operating, and you can sort of do a dive profile where you start deep and then work slowly back up to the boat over the course of an hour.
That's cool.
Yeah, so those are like highly productive oceanographic cruises where there's some science activity occurring during the night that sort of has a long timeline associated with it. There's the ROV dive during 8 to 12 hours during the day, and then a short, conditions permitting, a blue water dive in like midday. Yeah, and the remotely operated vehicle, it's in the water for 12 hours. There's a control room. Control room is like plastered with screens. Each screen is giving you a camera feed from the ROV, so you've basically got 360-degree eyes around this remotely operated vehicle. There's a captain's chair, right? There's a driver's chair. There's a scribe, so someone keeping notes on everything that's seen and what's being collected, what's not being collected, and entering it into the database, right? And then there's like the peanut gallery, and there's like rows of chairs where everyone is not directly participating in some function associated with running the ROV. We're all watching the screens and saying, oh, do we want to collect that? Or what is that thing? Closest I've ever had to what it would feel like to be on the bridge of like the Enterprise and Star Trek. I only got to drive an ROV once for a few minutes, and that was fine with me because that's high pressure.
You get to sit in the captain's chair?
Yeah.
Hell yeah.
With a little joystick to drive. The navigator's chair. But sitting in the science chair and sort of trying to keep up with entering any information associated with pictures that are being taken or video footage that are being taken that's a lot of fun.
That's
right, because everyone's chiming in about what they're seeing.
That sounds so cool. How do you think your experience training in this field, sort of a high adrenaline, high stakes, visceral environment, informs your approach to doing science?
I would say lack of fear of the unknown.
Okay, huge.
But that's something that all scientists have. Otherwise, they wouldn't be doing science. I suppose the most important thing is probably being calm when I have no idea what the outcome is going to be for an experiment, or even if it's like a physical activity. Not being worried about what the outcome is going to be, just being worried about what's happening in the moment. And as long as I can stay calm, then I can adjust as needed based on incoming information. If that makes sense.
It does. And that sounds like a life perspective almost, or an ability that can help.
Yeah, it comes directly from the experiences that I've had outside of science.
But then you bring that here, and you're able to do some some work that no one's done before, right? And then find some things that are going to add to what we understand about the world around us. And I think that ends up being why it's so important to explore how these different things we do shape our perspective, and then how we bring that perspective to asking big questions.
So I often think about trying to sort of simplify the human experience. You can think about how humans have increased their knowledge, right? So you've got some black box, and let's say that the black box represents all possibilities.
Yeah.
Totally ridiculous scenario, right? But let's just say that that black box is everything, right? What we know is some very thin, spirally thing moving through part of that black box. And so you can think about how we explore the unknown. How far can you get from that spindly bit of information that we know and understand what you're observing? The safest thing to do is to not go that far. You learn something new, but it's completely rooted in what was already known. Right? If you go a little bit further, right, it's a little bit more difficult to do, but it's also a little bit more difficult to explain to someone else. And if you can't explain, if you can't contextualize your observations, you're essentially not making observations, right? So you don't want to go so far away from what's known that you're a kook. Yeah. Right? Right? And then there are the unique individuals that go a little bit far away from what's known, right? Sufficiently far away that it's super interesting and you can see some path back to what is known in more than one way. So connecting bits of information from different parts of the human experience, those are the folks that get Nobel Prizes.
Well, Bill, from air to land to sea, you definitely have been all over. I want to thank you for taking the time to chat with me about your adventures, cool science you're doing, and I look forward to learning more about what you guys get into. So,
Oh, it was my pleasure.
Thanks for joining me.
Thank you.